Hello everyone and welcome to Protocol Design, what you need to know to ensure a successful study, presented by Roe. I'm Carly Helfand, editor at Fierce Pharma, and I'll be moderating this webinar. Our speakers today are Dr. David Shoemaker, Senior Vice President of R&D, and Dr. Karen Kesler, Assistant Vice President of Operations. You can read their full bios on the right side of your window. Just a few technical notes before we begin. If you have trouble reading a slide, please hold and drag the right corner of the slide window to enlarge. Please also disable your pop-up blocker to participate in the interactive parts of this presentation. If you'd like to download the slide deck, please click the purple globe at the bottom of your screen. This webinar is being recorded and will be available on demand within 24 hours after the event. We will follow the presentations by a Q&A session. Please submit your questions during or after the presentations using the Q&A box at the right of your window. Okay, now let's begin. Dr. Shoemaker, please go ahead.
Thank you. So today we're going to talk about protocol design and development, and I want to emphasize that this is really a nuts and bolts type of talk. So if you're looking for heady intellectual thinking, that's not what you're going to get here. You're going to get some down-to-earth basic advice on how to produce a successful protocol that will run a successful study. So these are just some quotes that I've collected over my more than 25 years of developing protocols. But I think the most disturbing one to me is the one at the bottom, which says that 70% of the money expended in drug development is wasted. And that was a quote from the former head of the European Regulatory Authority, the EMA. And I tend to agree with him based on the different poor quality protocols that I've reviewed over the years. And it's frustrating. And so this webinar is an attempt to bring everybody's best practices up to a ability to produce a successful protocol. So the first thing I want to talk about is commercial protocol development, where your goal is to get a product on the market to sell. And typically, these protocols are phase 1, phase 2, and phase 3, with phase 1 being focused on safety, phase 2 being focused on convincing yourself that your product works, and phase 3 being focused on convincing the regulatory authorities that your product works. But ultimately, in all the studies that you do, you should be collecting useful data that will support your marketing application. There are also a lot of people who develop protocols for scientific reasons, and they can be different scientific questions, whether to answer questions about mechanisms, mechanistics of particular drugs. They can be comparing different drugs. They can be trying to improve current therapeutic practices. But typically, these protocols are generated with the hopes of publishing the study results. They aren't focused on getting a product approved. And I'm not going to say a whole lot about these types of protocols in this talk. So by way of introduction, these are the topics that we're going to cover today. I'm not going to go through them one by one, but it will give you an overview of how I think it is best to design and develop a successful protocol. And then at the end, you see a path forward. Karen Kesler is going to join us and talk about what I think is a fairly common sense approach to developing protocols in the future. So, protocol development principles: state everything once. This is one of the most egregious errors that I encounter in protocols that I review. People pepper their protocol with repeated information, whether it be the dose, whether it be the frequency of the dose, or whether it be the patient population, ad nauseum. So it's a very inefficient way of developing a protocol. If things are stated once, you get it into the protocol, and you don't have to worry about editing it in 100 places. Another advisable practice is to write objectives first for early stage protocols, and write comprehensive and specific objectives for these phase 1 and phase 2 protocols. Whereas when you are developing a protocol with a lot of data and a lot of information regarding what you're going to do to approve the drug, you can write your statistical analysis plan first and develop the protocol after you understand where you are going in the study. And the final thing is to plan up front and avoid protocol amendments. I've worked with some companies who think it's just the goal is to get the protocol submitted to FDA as quickly as possible, and they are saddled with several protocol amendments that waste more time and a lot of money, but more time than if they had done the planning up front. So this is just an example of the number of individuals that should have eyes on a commercial protocol. This varies in terms of the time spent, but it helps to have each of these people review not maybe phase 1 protocols in all cases or phase 3, but you should consider these individuals when developing a protocol. As far as developing a protocol efficiently, and this is done by and large in the industry. When I started in the industry, people would just draft a protocol de novo, and so you were left with this 50 to 75 page document to edit right from the get-go. And fortunately, people have adopted this protocol synopsis approach, but some synopses formats are better than others. But if you can get an agreement on a 5 to 10 page protocol synopsis with the critical elements included, the protocol development ultimately becomes a word processing exercise. You're just taking the thinking that was put into the protocol synopsis and expanding it, so it's fairly simple. And if you have a lot of procedures around mixing the drug or administering the drug, please put those into a study procedures manual rather than in the protocol, because things like that through the development, through the trial, may even change within the trial. So you'll have to submit a protocol amendment. So get the details out of the protocol and into a study procedures manual. As far as early stage objectives, the type of objective that you're looking for is one that describes what you're doing in the study. I can't tell you how many early stage protocols I've seen where the objective is to determine the safety and efficacy of a drug. And it is just not worth anything to write an objective like that. So try to be as specific, such as the illustration given here, so that you know where you're headed. If you write a detailed objective before you begin drafting your protocol, the protocol synopsis actually becomes very easy to develop as well. So these are the elements of a clinical protocol. They are by no means unique. People call them different things. I would say that introduction and study design are pretty common as far as nomenclature, but these are the minimum number of sections that you must have in the development of a clinical protocol. And you do not need any more. You can put them in, but they're not required. So what I'm going to do is go through these different protocol sections by way of illustration. So objectives, we've started to talk about them. So if you look at objectives by phase of development, in phase 1, obviously, you're concerned about safety. Many times you're going into humans with a compound that you know virtually nothing about as far as previous human exposure. So you had best be cautious. And you're going to want to collect pharmacodynamic information, potentially, pharmacokinetics, and efficacy in some rare instances. Phase 2, the focus shifts to where you're trying to convince yourself that your product works. And so the primary objective will be efficacy. And you will be trying to ferret out what is the minimum effective dose of your product. But whenever you're injecting or giving your product to humans to ingest orally, you are evaluating safety. So every exposure is a safety number that will go into your NDA eventually. Phase 3, similarly, the focus is on efficacy. Now, the efficacy is an attempt to validate what you found in your Phase 2 program, and it's for the benefit of the regulators. But again, you're still exposing more subjects, so a secondary objective dealing with safety is mandatory. So, as I said, the quintessence of the early stage is focused on the objective. And it requires, as I said before, consultation with an interdisciplinary team. I think the first bullet are the mandatory people to get around the table initially to think about where you're headed with the program. It shouldn't be done flippantly. More often than not, what I find is a protocol is developed by pulling the last one that closely resembles what you're trying to do off the shelf and starting from that. And that is a recipe for a lot of wasted time, in my opinion. Early phase synopsis and protocol will flow from the objectives if they're written correctly. So this is just a diagrammatic of what I was talking about. The study objectives there in the center are the focus. You must collect some information before you can write valuable objectives. And from those study objectives, you can simply craft the protocol synopsis and protocol with very little effort. From the protocol, then you can develop CRFs and the database and everything downstream from there. In the objective, you must include, obviously, the product, the patient population, the generic dosing regimen, clinical endpoint, efficacy analysis, safety analysis, and phase, just so the reader gets an idea of where you're headed. Do not put the dose in here. This is another case of redundancy. A lot of people put the dose in the objective, and then I can't tell you how many protocols I've reviewed that have a different dose in the objective and in the study materials section. And where do you go from there? You have to go to an amendment if you've submitted it to the FDA and correct that error because it's a significant error. So state everything once. And this is just an overview of the product development process. I apologize, my voice is going. But it gives you some sense of how clinical fits into everything else that's going on in the product development process. So missed timelines in clinical can severely affect your overall development. Again, all of this is being done to get to a marketed product, and it must be coordinated. But clinical is but part of the overall product development process, and it needs to fit in. And so when you have delays in patient recruitment because you haven't done enough feasibility work to understand your patient population, everything suffers. So we move to phase 2. Again, this is the phase where you're trying to convince yourself that the product works, and this must be very detailed. Phase 2, I remember the head of the Office of Cellular and Gene Therapy speaking at a conference once, and he was saying that too many companies were moving from phase 1 and trying to guess the dose and do a phase 3 study. And he said that phase 2 was designed for a purpose. It is to find what is the minimum effective dose that doesn't harm the patients and yet confers efficacy. And so this must be a very well-crafted objective to define how you're going to examine dose. And this is just a diagram. You can use diagrams. It doesn't all have to be done in words, but this is a example of how one protocol was developed. In the top half of the slide, they're trying to reduce the dose to see if it works. And in the bottom half, the initial dose didn't work, so they're trying to raise the dose or raise the regimen, the frequency of administering the dose in order to get an efficacious result. And you need to think about things like this. This can be done in different trials, but these days I think everyone's started to use adaptive trial design such as this and answer a question within one protocol. But believe me, if you are planning on doing an adaptive trial design, it requires even more time for planning and thinking about the different choices that you have and the different results you may get. So it's not something to be taken lightly. So as far as data analysis by phase, phase one is pretty simple. You're focusing on safety. You're looking for treatment emergent adverse events. You're evaluating the pharmacokinetics of the product. Occasionally, you'll be doing drug-drug interaction studies and maybe compromised patient studies. But the statistics are not that difficult. They're largely descriptive and can be written fairly easily within the confines of the protocol. There's really no need for a statistical analysis plan for a phase one study. In phase two, however, you're starting to get more specific. You're starting to define successes. The focus, again, is on efficacy, and so you need to have a very good idea of what you're going to consider a success in order to go down to the FDA and have an end of phase two meeting. So, again, you're trying to convince yourself that you have a product, but your statistical methods ought to be rigorous enough so that you don't get down to the FDA and you realize that you've made a mistake. And then finally, phase three, these protocols are developed in tandem with the agency or with another regulatory authority. And there are plenty of studies in most diseases, not in rare diseases, obviously. Those are much more challenging. But for established diseases, there are precedents, regulatory precedents for what is a regulatorily acceptable clinical endpoint, and that is what you should use so that you and the regulators are on the same page. And then the statistical methods, again, in fact, in an end of phase two meeting, the best practice is to submit your phase three or your two phase three studies and get agreement from regulators on the design before you even begin. So that's objectives pretty much. Now we're going to move on to statistical methods, and if you have questions, I'm going to defer these to Karen Kesler because she is a biostatistician, but I will go through those I'll go through these points at a very high level. So again, this is the section that is the quintessence of the late stage protocol. It requires consultation. The statistician can write it, but either the statistician has to have a lot of regulatory experience and a lot of medical experience and a lot of scientific experience, or they need to access those disciplines in order to craft an effective statistical analysis plan. The SAP cannot be written very quickly. It can be, but the quicker you write it, the more chance you have that something's going to be missing. And once the clinical team has crafted the statistical analysis plan with the statistician, then with an understanding of that, the clinical database, the case report forms, and the protocol synopsis can be written. You can write all those documents ahead of time, but if the statistical analysis plan is not finalized, they may change. So it's up to you whether you want to do things in common sense, order, or if you want to continue with writing the protocol first and then retrofitting all the important information into it. And this is, again, diagrammatically just emphasizing the statistical analysis plan in the center is written with input from, obviously study variables from FDA, previous study design, but from that, the CRFs can be developed to capture data that is needed by the statistician to do the analysis. The database can be constructed, and after that is understood, the protocol can be written. So these are the basic elements in a statistical analysis plan. We've talked about efficacy analysis. Again, that's primarily phase 2, phase 3. I do not recommend overcomplicating your primary analysis or having co-primary analysis, as some people are wont to do. Figure out what's the most important thing. That's what primary means, and state it. And then you can have secondary analysis that can add supporting information. Safety analysis is, again, it's very important for the statistician to help you collect data consistently across studies. Too many times we're trying to construct NDAs from data that is all over the place, and you can't tell a story if you haven't organized your protocols correctly across the program. And obviously sample size and missing data are elements of the SAP. And these are some of the more detailed issues that we actually may get into in a later webinar, like group comparability at baseline, interim analysis, and stopping rules. I'm going to talk a little bit about stopping rules because they've become very prominent in protocol development these days. But these other analyses as well are critical for many protocols. So that's objectives and statistical methods. Now we're going to move on to the other sections, and I'm going to go through those much quicker and in a perfunctory manner because I think they are pretty well understood and not as complicated, and they flow from the objectives and the statistical methods. So the introduction, I want to emphasize brief. I review many protocols with 10-page introductions or 5-page introductions, and it is of interest to nobody. There is an investigator's brochure which has details, and you do not need to replicate the investigator brochure in the introduction to the protocol. So I emphasize brief. You do need a rationale for where the study you're doing fits into the overall development program. You do need a dosing rationale for each study you do. I've written BLAs with no dosing rationale, and believe me, after conducting 15 studies and not knowing what the dosing rationale is, it's a pretty bankrupt section that you write in your BLA. Don't just go with a dose that works. And similarly, study justification. Why is this study being done? And as the introduction proceeds through development, it should be shortened. There's no need to recapitulate animal data if you have human data. So the human data trumps animal data, so that is what should be summarized in your introduction. The toxicology is contained in the investigator's brochure. As far as a dosing rationale, this is incredibly important in phase 1, but it's derived almost entirely from your tox data and your no observed adverse event level in animals. And that needs to be laid out very clearly for FDA so that they feel comfortable with the starting dose that you're using in your phase 1 trial. Phase 2 studies tend to leverage the animal data less, and the human exposure data where you've done a single ascending dose study and maybe a multiple ascending dose study in phase 1, that's important for people to know the results of before you start your phase 2. And then eventually your dosing rationale is going to be based on the very comprehensive work you've done in phase 2, hypothetically. But you need to spend time thinking about it, and as I said, you're going to need to have this for the marketing application. So spend some time getting a dose that you feel confident is the minimum effective dose that minimizes the risk to the subject. And this goes in the introduction. You'll find this dosing rationale peppered all over a protocol. So I'm just recommending get it up front. As far as study design, I consider study design to be the quintessential dumping ground for information. It is unbelievable how long you will see study designs I've read have experimental procedures in it. They have time and events tables in it. It should be a very succinct section that flows from the objectives, and it should be limited to the design. So what is the design? Dose escalation, parallel, crossover. Is this an efficacy study? Is this a PK or PD study? What type of control are you using? Blinded? Is it single center or multi-center? But it can be written generally in 2 paragraphs. How long are the study periods going to be? How long is the study duration going to be? Again, don't put the dose here. And I've seen the dose 10 times in a study design section. And it's just a nightmare when you have to revise the protocol synopsis or protocol. So, as I said here, I know the Roman Empire crumbled in large part because their number system did not contain the number 0. Nevertheless, day 0 is verboten in protocol development. It is actually not part of CDISC standards. If people are familiar with CDISC, there is no day 0, and there is no week 0. It wreaks havoc with databases and in just talking about a study. So all you need is love and a time 0 on day 1. That works. Everything fits. There is a week -1. There is a week +1. Which week, I ask you, is day 0 in? Is it in week -1? Is it in week 1? Oh, it's in week 1. So you're telling me that day 7 is in week 2. That just is so counterintuitive that it bothers everybody who reads your protocol. But I think the most important thing is the CDISC standards, which are going to be required for all protocols developed after December of this year, has no day 0 in it. So please get that out of your protocol if you currently have it in. Study treatments. This is where you get together with your chemistry, manufacturing, and controls people and write this section. This needs to be very well written and understood by the clinician, the investigators, but it also needs to be chemically correct. And you need to get with your IWRS people for decoding procedures. And but this is not something that is written typically by one person. So be aware of that. And this is where the dose goes. Subject enrollment. This is, primarily the purview of your physician who is helping you develop this protocol. The inclusion-exclusion criteria should not just be copied over from the last protocol. It really is amazing the number of errors you find due to that practice. Randomization and blinding is described, and subject withdrawal, what's going to be done in the case of subject withdrawal. So inclusion-exclusion criteria, obviously in phase 1, you're going to usually be dealing with healthy volunteers unless you're working with a life-threatening condition. There's no real reason to go into patients in phase 1. It complicates matters. It's a very valuable thing to understand how your drug is handled pharmacokinetically in a normal person, with no disease ramifications affecting the PK. Then in phase 2, you can go on and optimize your population to help you demonstrate efficacy. It should be a skewed population where you enroll individuals to help you demonstrate a success in phase 2. And then once you're convinced that your product has potential, you broaden the population out in phase 3 to more closely represent the population that you will be giving your drug to in the population at large. Randomization and blinding, one-to-one is always preferred from a scientific perspective. However, 3-to-1 is always preferred from a monetary perspective. You can typically do several doses and include as many placebos in each of the doses so that you have equal groups with which to compare, and that is a valuable way to look at it. Double blind is preferred. Single blind is not as preferred, but in some cases may work, and open label is to be avoided at all costs unless there's no other way to do the study. Subject withdrawal, obviously you've got to have criteria for discontinuation, and you have to have a plan if you're going to replace dropouts. Not all protocols replace dropouts, so it's not required. But this is not where you put stopping rules, which warrants its own section. Study assessments. What is done when? And typically this is done by day. Visits, people talk about visits ad nauseum in protocols. For someone reading your protocol for the first time, visit means nothing. They don't know when you're going to come in for visit 13. That's not valuable. You must have a day. You can use visit as long as it's accompanied by the day of that visit. But to pepper your protocol with visit 1, visit 7, visit 13, they have to flip back to the time and events table every time to figure out when that's being done. You have study assessments also written in textual format besides the time and events table. The first thing I do, or not the first thing, but one of the things that I never fail to do when reviewing a protocol is to compare the time and events table with the textual format. And I would say in about 50% of the time, I find inconsistencies. And the reason that you don't want those inconsistencies is that one study coordinator at a site will be doing the procedures based on the time and events table, and a study coordinator at another site will be using the textual format. Consequently, you're going to get two different results from two different studies for the same protocol. Not wanted. Reporting adverse events. This is a section that should be written by your pharmacovigilance expert. And it should be done in, I think, compliance with the regulations, in as much as this is probably the most or the easiest way to get in trouble with FDA is to miss a SAE. So there are very rigorous reporting requirements that you must adhere to, but this section can be written once and then put to bed until there's an update in the regulations. It's not something that needs to be written every time. It can be written into a template and just pulled into the protocol for development efficiencies. Stopping rules. Again, this section is not always required, but it seems to be required more and more, especially with the evolution from, sort of blockbuster development to rare disease development, because not much is known about many of these rare diseases, and you're really in a difficult position as far as determining when to stop the trial. So it is a complex algorithm that needs to be developed with physicians and biostatisticians and regulators. And your goal is to convince the investigators that are doing your study and the regulators that are allowing you to conduct your study that you are proceeding in a safe fashion. It is a difficult section to write, but there are examples out there that you can use to help you design your algorithm. And I just give you some generic stopping rules here as a starter to give you some idea. Some stopping rules are paired with an interim statistical analysis. If you're heading towards or achieving futility, that's one reason to stop the study. Or if you're fortunate enough to have eye-popping efficacy, that is another reason. That doesn't happen in my experience, but a lot of people plan for it. So here's some suspension guidelines. It's a little bit, it doesn't require you to stop the trial, but allows you to obey the trial while you evaluate the data that's coming in, and it's meant to prevent stopping a study for an inappropriate reason. These are essentially stopping rules, but they're just, they've got a little bit more wiggle room and gives you some time to think about the results and the data that you're seeing. So data handling and quality, again, this is another section that can be written once, and until the regulations or the guidances change, can be put to bed in a template and then pulled into your protocol with no need to review. And in your draft protocol that you send out, you probably should have in parentheses, do not review. Otherwise, people are wasting their time reviewing these sections. And then finally, I just give you a list of the administrative considerations that need to be included in the protocol. I'm not going to go into these in any great deal of detail because there's not enough time, but these complete your protocol, and again, should be templated unless there's some recent changes to regulations or guidances. Similarly, with the investigator statement, it's important to find investigators that are committed. I leave that to you, how you do that, but clinical research is probably, the fifth priority of most investigators. And so somehow you need to get an understanding from them that they're going to conduct your study in accordance with GCPs, or all the work that you've done developing your protocol is for naught. And a study can be spoiled by one site and one investigator. And references, my only pet peeve about this is that some people feel like they're writing a scientific paper and put a footnote after every sentence in the document. And this can lead to hundreds of references in a protocol. And I think typically 20 references is a good number to shoot for, or less, because it's really not about people doing research. It's about common sense development of a protocol to form a successful study. So just some common pitfalls in protocol development. We've talked about initially drafting a complete protocol, not spending sufficient effort on the objectives or the SAP, submitting an SAP after a study has started. This is not always a death knell, but it's just not a good, healthy practice, and it requires changes potentially to all the documents that are developed without understanding what the final SAP is. Peppering a protocol with redundant information, not checking that the time and events table and the text of procedures for consistency, not soliciting sufficient interdisciplinary input, not identifying final decision-making authority, and allowing endless reviews. We have a terminology here that it refers to endless reviews as circling the moon, and some people are wont to do this. So I'm going to turn this webinar over to Karen Kesler now. She's a fabulous biostatistician, and she's going to talk about what I think is the future in, and she thinks is the future in, protocol development.
Thank you, David. So the solution and the future where we're heading is really to treat your data, treat your protocol as if it was data. Okay, I think the buzzword here is metadata. It's data, it's information about data. And I know that can be kind of a weird concept. We think of protocols as documents, not as databases. But even if you just take the title of a protocol like I've got here, when you look at it more closely, you can see that it's actually full of data. All of the colored highlighted pieces here are actually data, and it's just strung together with a bunch of prepositions and conjunctions. Okay, so here you've got what phase is it? That's a piece of data. What is your compound? What is the dose here? We've got what is, whether it's randomized, whether it's placebo-controlled, we've got all of this information in there. And this is just the title. Imagine how this could work if it was everything about your protocol. Now, luckily for us, some very smart people over at CDISC have been putting a great deal of thought towards this. For those of you who are not familiar with CDISC, it's a group of mostly volunteers. It's a nonprofit development organization, but they do a lot of work with volunteers from the industry. And they have standardized a lot of data structures. So data structures for sending to the FDA, which would be SDTM, data structures for analysis data sets, ADAM, and then even going back to case report forms, there's the CDISC initiative. Well, they've thought about this for the protocol too, and that is the protocol representation model. What they've done is they've taken all the standard concepts and basically converted those into fields. Okay, so you have things like your objectives or your phase, and what degree of blinding do you have? All the pieces that go into this, even information about your population. And then you have this structure whereby the data is common to every protocol you're looking at. Now, obviously not every study is the same, but how you capture that data and what the values can take on are common across the protocols. And why do we think this is the solution? When CDISC talks about it, they talk about it in a very dry format. It can be readily entered into an information system, or it allows it to be searched, analyzed, and whatever. Honestly, at the risk of sounding like a QVC commercial, there are thousands of uses for this data. Okay, it's incredible. You can automatically update your study summaries when you're posting to your websites. You can look at, you can transfer much more easily when you're data sharing, when you're going into EDC situations or into your CTMS. You can have the schedule of events transferred over into your EDC system, and so you've already got the structure for how many visits you have for your study. Just an example. Your sites are part of your protocol. If you capture that information as data, you can pass that through to your CTMS system, and you don't have to enter it again. You reduce errors. Everything is consistent. There is some very good work being done with searchable databases. I think if you have a lot of compounds and a lot of clinical areas, this can be huge. Where you go in and say, how many studies do I have in rituximab? Well, that's used across autoimmune, solid organ transplant, and several other clinical areas. And perhaps you wouldn't know that unless you could go and search for that easily. The most relevant piece to our talk today is that this matches what David was talking about in terms of the protocol development process. Okay? So you want to go in and identify these key pieces, your objectives, your endpoints. How are you going to analyze those endpoints? What does your population look like? And when you approach it from the perspective that those are data points, okay, that you need to define, it frees you away from going in and immediately writing text. Okay, and viewing it as a sentence. Instead, it's a data point. And I think it adds a certain layer of discipline to the process of developing your protocol. It's structured, you can go through it, and you can eventually, you can then fill it in. And on top of that, you can fill it in once. All right, so as you're going through your development cycle, and you change something, you change the dosing, for example, you can go in and change that in your database, and then it can propagate forward into your document, and it can go wherever you refer to that dose. Admittedly, if we're writing a good protocol, as David says, we should only have it in the protocol once, but you understand the approach here. And we do have that ability to take data and make it into a document. If we do it this way, it can get you 50% of your protocol written. And all you need to do is go in and add those prepositions and sentence structures. The other great thing about this is the CDISC people have been trying to make this easy. They've got some toolkits out there. They've got a lot of information about what the recommended fields they have, what is this protocol representation model, which is really the collection of data points, the definitions, what fields can go in, and they've got it all in one place in an easy-to-use thing. They're also looking at putting together a wizard. I like to think of that as the TurboTax approach to writing a protocol. I think that that's a few years off, potentially, but I do think we will get there. So in conclusion, it really takes a village to make a protocol, okay? You need to make sure you're getting input from the subject matter experts across the board. Developing a protocol also benefits from having structure, okay? Having a plan, making the decisions before you're writing. And if you have a well-planned protocol, that is the first step to having a well-designed and well-implemented study, okay? If you don't have a good protocol, there's going to be more confusion, there's going to be more problems. And as David has pointed out, there is no control experiment for writing a protocol. You just kind of have to go and do it. But the good news is there's a lot of help out there for it. So we want to thank you guys for joining us today, and we've got a few questions to answer. Oh, and we can hand that off now back to our moderator. Okay, so I think some of the questions that we have, one of the first ones is talking about, the statistical details and why those should be in the protocol and why we should develop the SAT, prior to developing the protocol. This goes back to what I have heard is called the tables first approach, where you decide what the answer is that you're after. You get disciplined about what the question is that you are trying to answer with your study, okay? And then the second step to that is, what would my displays look like in trying to answer that question? What do I need to cover? And then you back off of that and say, okay, how do I analyze that? How do I get to those statistics, those displays that I want? And what we're trying to do here is ensure that we think through the process of how we would define and analyze all of the data that we're collecting in the protocol. Because how you define it in the protocol is how you're going to collect it throughout the study. Now, I don't know that you necessarily need to have the SAP completely finalized prior to the protocol definition or finish, but it reflects a thought process, and it forces you into that thought process and makes sure that you got all of that straight before you put it in the protocol.
And I just want to add that a good portion of protocols that I review, as Karen said, they've actually thought about what they want their results to be. Unfortunately, they're assuming that their product is going to work. So the objectives and the statistical analysis plan are actually biased, and that is probably one of the quickest ways to, have an unsuccessful protocol. You might get lucky, but you should always go into protocol development with the objective of determining whether or not your product works or it doesn't. So you should be evaluating efficacy. You should be evaluating safety in an unbiased fashion. You should not be determining efficacy or counting on efficacy of your product, which is written into some protocols, perhaps subliminally, but nonetheless, that's the perspective that you get when you read the protocol.
So our next question is why oncology and HIV studies do phase 1 studies in non-healthy, in affected individuals? And certainly for oncology, that has to do with the therapies available. Chemotherapy is very toxic, and it was determined that you don't want to expose healthy individuals to something going into it is going to be toxic. So those are often done in late stage cancer patients.
Yeah, and I think actually in the last 2 or 3 years, there's been a huge advent of immunotherapy in oncology, so that's no longer the case. You can sometimes do normal volunteers trial with oncology products, but back 5 years ago, most of the oncology therapies were chemotherapeutic compounds that were actually noxious to normal volunteers, and you just couldn't do a study in normal volunteers.
Another question along those lines is, what are some of the challenges associated with working in an oncology protocol development scenario, especially lung cancer, I think it was talking about?
Well, I think oncology is very different from virtually any other type of product development. It's a very difficult population. Lung cancer is typically a very seriously ill population. So it's difficult to understand all the side effects that you're going to have in the context of a very sick individual. So it's difficult to parse out what is due to your product and what is due to the progression of the disease. And there actually was just a guidance released by FDA last month on the formation of safety assessment committees as an idea to help with these sorts of protocols where you predict a priori what are the adverse events that you anticipate to occur in your patient population, and that with a safety assessment committee comprised of several physicians and statisticians, you can make an interpretation of the trends that you are seeing in real time, and that allows you to basically develop oncology products more safely.
Thank you to both of you. I'm back after a technical difficulty. Just a reminder, everyone, to please submit your questions using the Q&A box at the right of your window. We've received a lot of great ones already, and we're trying to get through as many as possible. So the next question is, do you recommend submitting your protocol to your FDA reviewer for analysis and feedback prior to beginning a trial?
Absolutely not, except if it's the adequate and adequate and well-controlled phase 3 studies, which you would submit in your end of phase 2 meeting package. You should be able to access enough intellect and experience without going to FDA. FDA is not a drug development advisory board. However, for the phase 3 studies, yes, you have to submit. I submit final protocols in the end of phase 2 meeting package to get FDA concurrence before you begin those, because the purpose of those studies are to convince the regulators that your product is efficacious and safe with a reasonable risk-benefit ratio. So in that case, yes, but otherwise, absolutely not. Once you open your IND, you are off to the races, and you are responsible for collecting enough experience and intellect in the medical and statistical and scientific community to design a viable protocol for a study.
The next couple of questions. Sorry, go ahead, Carly.
That's okay. The next question is the DSMB required for all phase 1 studies?
No, it is not. And actually, it has less to do with the phase of the study and more to do with the population. If you have a high-risk population, such as a pediatric population, or one where they are very sick, you may want to consider utilizing a DSMB in those cases. It's also good for situations that are high risk, like a dose escalation study. You may not need a full DSMB for that, but you'll definitely want some kind of independent medical monitoring. If there's it's really all about the risk to the subjects. And if you perceive for whatever reason that there's a higher risk to the subjects, you should definitely consider a DSMB.
Great. And then the next question, if there's going to be an independent data safety monitoring board, what needs to be included in the protocol regarding the structure and function of the DSMB?
It definitely does need to be in the protocol that you're going to be using a DSMB. But that's pretty much it. We would definitely not want, you don't want to get into a ton of detail, okay? You want to give yourself the flexibility if the members have to change. You don't want to put the members in, a lot of the information that you're going to be giving the DSMB also changes, so you don't really want to put in a ton of detail about that. The mere fact that you have the DSMB and potentially how often they're going to be reviewing data is about what you need in the protocol.
Similarly to what we were talking about earlier with the preparation of the drug, a lot, if not all, of the information about the DSMB is contained in the DSMB charter. So simply referring to that in the protocol is sufficient.
Great. And for the next question, in an oncology trial, can you explain the relationship on investigation drugs, standard therapies, and supporting therapies?
I'm sorry, could you repeat the question?
In an oncology trial, can you explain the relationship between investigative drugs, standard therapy, and supporting therapy?
So standard therapy would be what the patients would normally be on, okay? So if you've got an adjuvant therapy, standard therapy is relevant there because that's the standard of care. An investigation drug is your new compound that you're adding to this. And then supporting is whether or not it's actually treating the cancer or whether it's something that's going to support, an adjuvant. It's going to make the patients feel better. It's, reducing nausea, for example, or whether it's going to, be an add-on and somehow make the chemotherapy or whatever the investigational drug is supportive. So that's kind of the gist there.
And can you explain how Phase 2A and 2B work within the Phase 2 protocol development process?
Any way you want it to. So I will elaborate. Some people refer to Phase 2A as single dose. Some people refer to Phase 2B as multiple dose. Some people use other terminologies. It's all Phase 2 to me. I've never used those letters, and it's never gotten to me any trouble with FDA, and frankly, I think it's more confusing. A lot of people, and this is where I see it used more than not, is that they'll do a Phase 2 study, and because it's their first Phase 2 study, they will call it Phase 2A. And subsequently, because they want to convince people who are funding their program, they want to demonstrate progress. So abracadabra, their next study is Phase 2B, which to me makes, little sense in product development.
And then this will be our final question. We had a lot of great questions today and couldn't get to them all, but we'll be getting back to everyone who submitted personally after the webinar. Can you comment on protocol development for digital mobile apps?
So protocol development, is this team effort. And I'm not sure about the digital mobile app aspect of it. If the question is more in regard to what do we need to put into our protocol when we're using apps, like, for example, to collect patient-reported outcomes, here again, it's like any other piece of data. You need to put in what you're collecting. If it's an important secondary, you need to put in what how you're going to analyze it and what some of the quality measures are there. But I don't think you necessarily need to put in a ton of detail about the device, the app itself. As long as you're referencing it and people can go get that, you could consider that as an appendix or a standalone document where you're, like a manual of operations or something like that. But I really think that it's actually going to be very similar to central labs or some kind of, mechanistic thing. You want to go for enough information that people can understand what you're collecting, but not too much.
All right, well, thanks to everyone for attending this Fierce Live webinar and submitting so many great questions. I'd like to thank our speakers for participating and Ro for presenting today's webinar. This webinar has been recorded. You'll be able to access the recording within 24 hours on the same page you used to register for this event. Thank you again for joining us, and we look forward to seeing you at future events. Hello, everyone, and welcome to Protocol Design, what you need to know to ensure a successful study, presented by Ro. I'm Carly Helfand, editor at Fierce Pharma, and I'll be moderating this webinar. Our speakers today are Dr. David Shoemaker, Senior Vice President of R&D, and Dr. Karen Kesler, Assistant Vice President of Operations. You can read their full bios on the right side of your window. Just a few technical notes before we begin. If you have trouble reading a slide, please hold